In this lesson, we're going to look at cultural microorganisms, so growing microorganisms or bacteria in a lab. Now, this is a Petri dish. Now, inside the Petri dish, there is agar jelly, which is a culture medium. So it's going to provide the bacteria with anything that it needs to grow. So, for example, carbohydrates, minerals, proteins, vitamins, so anything it needs to grow. Now, bacteria will form visible colonies. If I just show you another Petri dish. Now, as you can see on this Petri dish, all this bit here, now those are visible colonies of bacteria. So this Petri dish has been left for a few days and the bacteria have grown. Now in schools, this Petri dish will be left at around 25 degrees. You don't want to keep it any higher than that simply because you don't want harmful pathogens to grow. Whereas in industry, where you want bacteria to grow very fast so that you can utilize it, the Petri dish will be kept at a higher temperature. Now I'm going to investigate the effect of different antiseptics on bacterial growth. So here's my Petri dish from before that I'm going to use. And here are the 4 different antiseptics that I'm also going to use. So I've got alcohol gel, Milton, Virkon, and antiseptic disinfectant. The bacteria that I'm going to use is B. subtilis. Now before I start, I'm just going to turn the Petri dish over and divide it into 4 quadrants. Just going to use a ruler to help me do this. I'm dividing it into quadrants so that I can label the antiseptic. So AG will be alcohol gel, M for Milton, V for Virkon, and then AD for antiseptic disinfectant. And I won't get the antiseptics mixed up. To transfer the bacteria, I'm going to use an inoculating loop. It's just a long stick with a tiny loop at the end. Now, it's very important that I use aseptic technique. Now, that means I'm going to try and prevent contamination by unwanted microorganisms that can potentially affect my results and result in the growth of pathogens that are harmful. So the Petri dish and the culture medium, so the agar jelly, have been sterilized before use. So that means they've been heated to a very high temperature to kill any unwanted microorganisms. And because I am using an inoculating loop, I'm going to make sure I sterilize it as well. So at the side, I have got a Bunsen burner on. And every time I transfer the bacteria onto the agar jelly, I am going to just pass it through the hot flame. I'm just going to heat my inoculating loop to sterilize it. Wait a few seconds to cool it down so I don't burn the agar jelly. And then just put it into the bacteria and lightly spread it. To ensure there's an even spread of bacteria, I'm just turning the Petri dish around so that I can spread the bacteria. I'm just going to pop the lid on. Now, in order to transfer each paper disc onto this Petri dish, I'm going to use a different set of tweezers which are in this sterilized pack. So I've got 4 different set of tweezers. It's important to use different tweezers so I don't cause cross-contamination between the results. So I'm just going to start off, grab a paper disc. So I'm starting off with Virkon. Just going to place it into the quadrant which is labelled Virkon. Now remember, I'm going to use a different set of tweezers. Antiseptic disinfectant. Alcohol gel, and then last but not least, I've got this one that's left. As you can see, I have used tape to seal the Petri dish. This is to prevent any microorganisms from the air entering the Petri dish. Now, I'm not going to store the Petri dish this way. Okay, I need to store it upside down. So the agar jelly is at the top and the lid is at the bottom. So this is to prevent any condensation from falling onto the agar. Okay, so I'm going to leave this Petri dish for about 48 hours at around 25 degrees Celsius. Remember, I don't want any harmful pathogens to grow. Here we've got a different Petri dish. Instead of using B. subtilis, I have used E. coli for the bacteria, and I've used 3 different antibiotics. So I've got antibiotics S, C, and D. As you can see, there are rings around antibiotics S and C, but nothing around antibiotic D. So we're just going to ignore antibiotic D for now. The rings that you see represent the inhibition zone. So this is the clear zone around the paper disc where the bacteria have been killed. So the larger the inhibition zone, the more effective the antibiotic is. And we can see that antibiotic C has got a larger inhibition zone, so therefore is more effective. But in order to be more accurate, we can calculate the area of the inhibition zone using the diameter. So just going to turn this over. What you need is a ruler, and you need to measure the diameter of both inhibition zones, and you are going to measure in millimeters. So just to remind myself, the small one is S. Now I got approximately 18 millimeters for the diameter. For antibiotic C, I got approximately 30 millimeters. To calculate the area of the inhibition zone, we need to do pi, which is 3.14, times the radius, which is half of the diameter squared. So the diameter of S was 18 millimeters, half of that is 9 millimeters. So the area of S is going to be pi times 9 squared, which is pi times 81, and that gets you 254 millimeters squared for the units. And then for antibiotic C, the diameter was 30. Half of that to get the radius is 15 millimeters. And then to work out the area, you do pi, which is 3.14, times 15 squared. 15 squared is 225. So 3.14 times 225 gives you 707 millimeters squared. So we can see that the inhibition zone around C is approximately 3 times larger than the inhibition zone around antibiotic S. As the area of antibiotic C is nearly 3 times bigger than the area of antibiotic S, we can say that antibiotic C is nearly 3 times more effective than antibiotic S.